Right. Hi, everybody. Thank you for coming out to the final talk before the break here. My name's Ahmed Hasan. I'm a PhD candidate at the University of Toronto, and I'm excited to be here. I'm grateful to Professor Gaucher for inviting me to the symposium, and yeah, I'm excited to share some work we've recently done on how salt stress affects mutational input during the process of adaptation. So to begin with, it's probably no surprise to most people in this room that mutation rate varies at multiple levels in nature. So for instance, we've seen variation across the genome as seen here in this distribution of intermutation distances. We've observed variation between individuals as far as mutation rate is concerned, and so on and so forth between species and even taxa. And although that seems, I'd say, broadly well appreciated, perhaps something that's a little less well appreciated is the fact that environmental stressors can also affect mutational input. So just some recent examples of this principle. A recent study showed that in Daphnia, copy number variant rates can vary between different metal stressors. So those colored bars on the top left of the figure here show about twofold variation in copy number variant rates between different metal treatments, between nickel, copper, and so on. And of course, in everyone's favorite model plant, Arabidopsis thaliana, a paper in 2014 showed that indel rates are actually far higher in saline conditions, about fourfold higher in this case. And this is all well and good. If we're interested in mutation rate, this is cool in of itself. But the fact of the matter is that these stressors are often themselves the source of selection pressure as well in natural populations. They might impact spontaneous mutation, but if we're to consider some sort of circumstance where we have individuals trying to adapt to these stressors, then as it stands, any novel genetic variation that's now introduced, that's made available to selection, that sort of mutational input can be affected by the very source of stress itself. The way we're interested in approaching this question is through salt stress, in particular, in the freshwater alga Chlamydomonas reinhardtii, which is what our lab uses. So reinhardtii is a haploid single-celled individual, and in this two-panel figure here, on the left-hand side, we have reinhardtii cells in sort of benign medium with no salt whatsoever, whereas the cells on the right are in 0.2 molar salt. And although they kind of look fine in this still picture, much like many grad students, they might be quite stressed. In fact, salt stress actually affects growth more directly by disturbing the balance of ions and causing sodium toxicity. If we're to look at how salt stress over sort of a sliding scale affects reinhardtii, it's perfectly content growing in medium without salt whatsoever. But as soon as we up the salt concentration to about 5 to 7 grams per liter, the growth rate is inhibited by up to 50%. If we up that still to about 10 to 11 grams per liter salt, which is what we have in that picture on the top right over there, photosynthesis is almost completely inhibited. So like I said, quite stressed. And just to put these values into context, the average salinity of seawater, and I did mention reinhardtii as a freshwater alga, seawater has an average salinity of about 36 grams per liter. So as it stands, given how reinhardtii kind of struggles perhaps getting up to here, this sort of marine seawater way of life seems quite inaccessible to it. So if we were to have reinhardtii cells adapt to higher salinity environments, we'd also have to perhaps ask ourselves, how does salt stress influence mutational input along the way? Another way of restating this question is, how does salt stress affect the rate and distribution of new mutations that arise? Perhaps we could compare this with the rate and distribution of mutations that are retained during adaptation to salt stress. And then finally, by perhaps comparing these two mutation sets, we can then think about what this tells us about the process of adaptation to the stressor and how stress affects spontaneous mutation along the way. To be clear, a quick disclaimer, what this is not, we're not trying to profile the genetic basis of adaptation to salt stress yet. That might be for a future study, but this is more about mutational input, genetic variation during the process of adaptation and any biases that stress might introduce for now. So our experimental setup involves 3 separate lines. The first is a mutation accumulation experiment conducted in what I'll refer to as benign growth conditions, so 0 grams per liter salt. A quick reminder about mutation accumulation experiments is that they essentially involve bottlenecking lineages over many generations to just maximize the strength of drift, minimize the strength of selection. And in doing so, we're able to get a pretty solid picture of what spontaneous mutation looks like in the absence of selection, since unless mutations are lethal, any mutation has a chance to fix given this repeated bottlenecking. So this mutation accumulation experiment was actually done and published by my advisor in 2015. So this data has been around for a while, but what we're adding to the study is another mutation accumulation experiment, this time in a fixed high salt concentration. Specifically, it's another mutation accumulation experiment, but this time all the media are supplemented with 5 grams per liter salt, which, if you remember, inhibits the growth rate by up to 50%. The final piece of the puzzle, the final set of lines we have here, underwent actual selection in increasing salt concentrations. They're salt-adapted lines, more or less. So we started with strains in the lab, and over the course of about 300 generations, we increased the salt concentration to about 30 grams per liter salt. Most cells died, but 2 lineages that we've since termed SL1 and SL2 were able to survive up to that incredibly high saline concentration. Just to try to extend the limit of salt tolerance a bit, flowing across with another strain, CC2935, that was not included in the original strains, we were able to obtain lines, henceforth known as the high salt or HS lines, that have been surviving in the lab at 36 grams per liter, at this sort of seawater level of salinity. So what we have here with this setup is, 36 grams per liter. But what we have here in the setup is, from the original MA experiment, we see the spontaneous mutation profile. We now also have the spontaneous mutation profile under salt stress, or what to expect out of that. And finally, a set of mutations that were actually retained after selection, actually retained after lines were adapted to high salt conditions. So looking back at our questions here, we're interested in the rate and distribution of new mutations here across these 3 sets. So starting off with the mutation rate, I've got 2 panels here. On the left-hand side, we've got single nucleotide mutations and then indels on the right across our 3 data sets, with mutation rate, of course, on the y-axis. And off the bat, what we see might not be entirely unexpected. We see more single nucleotide mutations than indels across the board. But what sticks out here, if we look closer at the right-hand side panel, is that in both salt conditions, there seems to be an elevated indel rate. In the orange, the HS, the salt-adapted lines, more indels were retained than, even in the original MA. And then the salt MA lines in particular seem to have an incredibly elevated indel rate. So this piqued our curiosity. We decided to dissect it a bit further by looking at indels within genes and outside of genes. And what we see looks something like this. On the left-hand panel, so this is just the indel rate. On the left-hand panel, we've got the indel rate in intergenic sites, and on the right-hand panel, in genic sites. And what we see is that well, there are a lot of indels in both salt conditions, and as far as intergenic sites are concerned, the indel rate is essentially almost the same. But in genic sites, far fewer of those indels are actually retained. The takeaway here is that salt seems to elevate the indel rate, like we saw in Arabidopsis earlier, but this time around, genic indels are likely being removed by selection, obviously with frame shifts and such being quite deleterious, as one can imagine, in genes. So this is an example here of how salt stress is modifying or potentially affecting what mutations are being introduced as lines adapt, but then selection, of course, is doing its thing and removing deleterious variants. Looking further at the distribution of mutations across the genome, perhaps the classical way to do this, first of all, is to look across annotations. So we've got various genomic annotations laid out on the x-axis here, and then the proportions of mutations on the y. This time around, we don't actually see that much in the way of differences between annotations. There's a slight difference between introns, but overall, no real genome-wide trends to write home about between our 3 mutation sets. That said, there's multiple ways to look at the spatial distribution of mutations. Specifically, we could also look at the overall genomic distribution in the form of intermutation distances, like I mentioned very early on. So we could ask, are intermutation distances significantly different between our 3 conditions? Because looking at it this way could reveal spatial clustering trends. If there's if there's a set of really small intermutation distances, that's likely because there's clustering of mutations occurring somewhere. So here in this plot, I'm going to show the same data in 2 separate ways. On the x-axis, I've got intermutation distances on a log 10 scale. The top will have cumulative density functions, and on the bottom plot, it's the same data but binned to make it perhaps a little easier to parse. And that ends up looking something like this. The first takeaway we see here is that if we look at the orange line up top, representing the HS, the salt-adapted lines, there seems to be an elevation in lower intermutation distances, indicating that mutations are actually closer together. When we peek a little bit closer at this, specifically by looking at the bins at the bottom there, or at the bottom left, I should say, we found that the HS mutations were actually clustered within 10 base pairs, significantly more than in the MA lines, but not significantly more than the salt MA line. And I mean statistical significance, of course. And this suggests possibly a role for multinucleotide mutations being generated by salt stress. Perhaps when lines are exposed to salt, multiple mutations are arising at a time, and then selection is perhaps able to move across the fitness landscape a bit faster with this introduction of novel variation in that form. We also noticed that 12 out of these 15 10-ish base pair clusters happen to be in genes, which was pretty neat. And I've mentioned that this study is not about profiling the genetic basis of salt stress, but lately our lab's been on a bit of a network bent. So we thought, hey, maybe we could look at something, look at these genes in light of a co-expression network. And we could look at mutated genes directly, look at Go annotations and the like, but doing so, we figured, kind of ignores any functional relationships between these genes. And lucky for us, a co-expression network in reinhardtii was published not too long ago. And here are 4 depictions of the same network on the right-hand side over here. So we thought, okay, network analyses might allow us to ask questions about how related these mutated genes are overall. Not just the genes with clusters, but genes that happen to retain mutations during adaptation altogether. Quick plug here. My advisor, Rob, is going to be giving a talk specifically about predicting mutation fitness with networks. That's in room 21 over there at 5 p.m. It's pretty neat. You won't want to miss it. But a quick primer on graph or network theory, for anyone unfamiliar. Networks consist of nodes, which in our case would be genes, connected by some number of edges. In the context of a co-expression network, if 2 genes are connected by an edge, that's likely because they're significantly co-expressed. What we're interested in here is calculating the shortest paths between genes that happen to retain mutations in the HS. In doing so, we're able to quantify how close these mutated genes are in the co-expression network, and perhaps that could yield something interesting. So what I'm going to show here is the distribution of shortest paths between mutated genes in the HS. There were 85 genes that retained mutations in the salt-adapted lines, and their distribution of shortest paths with one another, or minimum shortest paths, I should say, looks something like this, where on the x-axis, the minimum shortest path seems to range from 0 to 3, where 0 indicates 2 mutations in the same gene, 1 indicates mutations in adjacent genes, and so on and so forth. So in light of that, we randomly resampled 85 genes from the full network and from MA genes 100 times. And those distributions actually look something like this. They're much more normally distributed. They also look quite like one another. And what I'm really interested in are these differences at 0 and 1, where we see there's a lot more in the way of the HS genes. We could also look at the exact same data, or we could overlay these 2 distributions, the MA and the HS, and calculate differences at each bin. And that looks something like this. It's the same data once again, just differences between each bin. And we see here primarily there's a big elevation in shortest paths 0 and 1, indicating that mutations are in genes closer together, in the co-expression network, which could open up cool targets to look for the actual genetic base of salt stress, among other things down the line. So let's bring it all back together. Earlier we discussed how environmental stressors are a source of selection pressure and can influence mutational input. These results show that in reinhardtii, the rate of indel mutations was elevated under salt stress, but many of these indels were removed by selections as lines actually adapted to higher and higher salt concentrations. Furthermore, we found that mutations are closer together spatially in both salt conditions, salt MA and the HS, which suggests that salt stress might be introducing multinucleotide mutations, which may then subsequently be retained. And finally, more as a cool methodological aside, we found that when overlaid on the reinhardtii co-expression network, genes mutated during adaptation to salt are actually closer together than expected, which could be a cool starting point for a future study. The overall take-home, though, that I'd like to leave you with is that it turns out describing environmental effects on mutation can really help tease apart the effects of environment and selection of polymorphism. If we looked at the HS directly, we wouldn't have had these insights about indels or possible multinucleotide mutations that we did otherwise. So with that, I'd like to thank my advisor, my committee, my lab, as well as our funding sources. And I'm happy to take any questions, or not, I suppose. But thank you.